Good afternoon. I'm speaking from my office at the National Institute of Mental Health in Bethesda, Maryland. I am so honored to be receiving an award named in memory of Pat Goldman-Rakic. Pat was an inspiration, a role model for many of us, myself included, and this has been true throughout my career, really. This award holds special meaning for me because Pat was a member of the Laboratory of Neuropsychology here at the NIMH, which is the same lab I now head, and that's before she moved to Yale. And in fact, our careers overlapped briefly here at NIMH. Of course, Pat was known for her seminal studies of the role of prefrontal cortex circuits in cognitive functions. She examined how dysfunction of these circuits affects behavior, and she did this mainly with animal models, and especially non-human primates. And then she considered how dysfunction may lead to a variety of brain disorders in humans. These are my interests as well. One topic I've been interested in is one area of cognition is value-based decision-making. This requires setting up representations of value in the brain and linking those representations to objects in the environment. And these representations, in fact, guide much of our behavior. One of the key players in value-based decision-making is orbital frontal cortex. At the bottom are ventral views of the human brain at the left, a macaque brain in the middle, and a lateral view of a rodent brain at the right. And you can see the shaded region covers orbital cortex, and these shaded regions are exploded at the top so you can see more detail. I hope you can make out these little numbers. These are the parcellations of the cytoarchitectonic areas identified by Joel Price and his colleagues. And they're really just 2 points. One is all these blue areas are granular and dysgranular orbital frontal cortex that are found only in primates, not other mammals. Second, as you can see, almost all of these areas have have been identified in both macaques and humans. So whatever we might learn in macaques may well inform the human condition. And here we see a phylogenetic tree of the Uarchontoglires. The Uarchontoglires is the last common ancestor, or this is the lineage coming from the last common ancestor of rodents and primates. And these red bars show 2 of the frontal cortex areas most intimately involved in value-based decision-making. The granular orbital frontal cortex, which I'll focus on today, and the ventrolateral prefrontal cortex, or VLPFC, which I won't have time to talk about today. The granular orbital cortex emerged about 55, 60 million years ago, actually based on recent evidence a bit earlier. And the ventrolateral prefrontal cortex, as well as other areas that you might be familiar with, such as the dorsolateral prefrontal cortex, emerged later in anthropoid primates. In fact, interestingly, it was Pat, together with Todd Preuss, who first proposed in 1991 that the granular frontal areas were specific to primates. This is our subject, a macaque monkey, and the photo resonates with the topic of my talk, which is how do we identify and select goals for action? And this is very much at the heart of value-based decision-making. In the work I'll talk about today, we use selective cerebral lesions, disconnections, reversible inactivations, which is another technique pioneered by Pat. And in addition, we use physiological recording of neurons combined with causal manipulations to help understand how the circuits underlying behavior operate. So we've been studying orbital frontal cortex for many years, and in the next few slides I'm going to be showing you some data from 2 different experiments collected about 10 years apart. These were studies carried out by Alicia Izquierdo, who's now at UCLA, and Pete Rudebeck, now at Icahn School of Medicine at Mount Sinai. And they both involve studies of monkeys that received lesions of the orbital frontal cortex, shown here in this cartoon on the left. And on the right, you can see images from a group of monkeys, representative from a group of monkeys that received aspiration lesions of the orbital cortex, which is a mechanical removal of tissue, and other monkeys, another group that received excitotoxic lesions of orbital cortex. And you can see this white hypersignal is the location of injections of excitotoxin, which very selectively remove cells from the orbital cortex. And I feel comfortable directly comparing data here because the monkeys had the same lesions, the same boundaries of the lesions, and they were tested using the same behavioral test procedures. So one of the tasks in our battery was object reversal learning. This is a classic task, really a benchmark task, known to be affected by orbital frontal cortex damage in humans, monkeys, cats, and rats. So we really included this in our battery, as I said, as sort of a benchmark task. In this task, monkeys learn to approach a rewarded object and to obtain the food reward hidden underneath, and they avoid the unrewarded object. It's very simple. When the monkeys have learned this task, we reverse the reward contingencies. So now monkeys need to approach the previously unrewarded object. And the monkey's given time to learn this new discrimination. And when they've learned it, we reverse the stimulus reward contingencies again, and so on for several serial reversals. And the data I'm going to show you here are summarized on the right, has been known for a long time. So this was not a big surprise. Monkeys with aspiration lesions of the orbital cortex were impaired in object reversal learning relative to controls who scored many fewer errors. However, our monkeys with excitotoxic lesions of orbital frontal cortex were unimpaired. This was really quite shocking. We searched our souls. We didn't want this result. We didn't expect this result. But after doing additional experiments to ascertain, this is, in fact, a clear and true result. So we think the difference between the aspiration lesion and the excitotoxic lesion is that the aspiration lesion disrupts fibers of passage, that is, fibers going nearby or through orbital frontal cortex, which the excitotoxic lesions don't. So this overturned dogma from decades that have been known since the 60s, from the time of Charlie Butter, that orbital frontal cortex damage was associated with reversal learning, and we've overturned that. So we turned to another task called the devaluation task. Really, we wanted to test the idea that was already out there, that orbital frontal cortex might be important for representing value of outcomes, literally the value of foods and fluids that might be rewards or items that you might want in the environment. And that representation would also contain the information about the sensory properties of those foods and fluids. So this task is, devaluation is a bit of jargon, but what the monkeys are required to do is, just as was the case for object reversal learning, the monkeys need to discriminate objects. Unlike reversal learning, however, they need to link objects with food reward value. And we know this because we ask monkeys to learn about particular objects here designated A and B. Some objects are associated with one kind of food, say peanuts. Other objects are associated with a different food, say M&Ms, and then we manipulate the value of that food and see how it affects monkeys' choices. So there are really just two phases. There's a training phase in which monkeys learn about a large number of objects and their associated food rewards. So some objects are associated with peanut rewards, as shown here. Other objects are associated with M&M rewards, as shown here. And when the monkeys learn those and learn about those objects, we bring them to a series of probe tests. And here, for the first time, monkeys are allowed to choose between two rewarded objects. And for 2 sessions of baseline probe tests, we simply allow monkeys to choose between peanut objects and M&M objects, and we record those choices. Those are our baseline choices. On a separate day, monkeys are pre-fed one of the two foods, say peanuts, in an attempt to devalue that food. And then we bring the monkey to the test apparatus and again allow them to choose between these two classes of objects. And then finally, on yet another day, the monkeys are pre-fed the other food, brought to the test apparatus, and so on. So intact monkeys do a clever thing. If they've been pre-fed one food, say peanuts, at the time of these choice tests, they avoid the objects overlying the sated food in favor of the remaining object. And we can quantify this shift in choices as a difference score, which is what you see on the next slide. So here are data summarizing this study. The white bars are scores of control monkeys, and the difference score here really means that monkeys are shifting almost 20 out of a possible 30 choices. So this means they're doing, the higher the bar, the better the job the monkey's doing, shifting their choices away from objects overlying the food, overlying the sated food. The color scheme is the same as the last data slide I showed you. So here we see that monkeys with aspiration lesions of the orbital cortex shown in red, as well as excited toxic lesions of the orbital cortex shown in green, are significantly impaired relative to controls. And in this case, it seems to be the case that we can say for sure that it is cell bodies of the orbital cortex that are producing this impairment. So these data are telling us that monkeys, first, our control monkeys, care about the value of the goal. They shift their choices if the value of the goal has changed. And this is the kind of behavior that underlies goal-directed behavior. And it's also telling us that monkeys need their orbital frontal cortex to link objects in the environment with current value of the food. What I haven't stressed is that on these probe tests, there were a different pair of objects on every trial. So there's no opportunity for the monkey to relearn the association between an object and the current value of the food. Instead, the monkeys are making these decisions on the fly. So this is very much a mechanism that allows monkeys to make good choices without having to incur additional trials and errors. And one more important piece of information is that all monkeys operated in controls alike, if offered the food, if offered choices between a sated and unsated food, will all choose the non-sated food. In other words, they don't want the sated food, so they can visually recognize the sated food. They don't want the sated food. So this impairment is very specific to linking objects with the current value of the food. You might think there are these alternative interpretations that I should consider. I can tell you my colleagues and I have investigated these possibilities and we can rule them out. So again, our monkeys have intact visual perceptual abilities, motivation, that is, they're just as willing to work, just as hard as intact monkeys to earn food rewards. Their food preferences have not changed. And again, our monkeys with orbital cortex lesions have intact satiety mechanisms. So this is a very selective impairment. Of course, the orbital cortex doesn't work in a vacuum. We've determined from disconnection experiments, experiments which allow us to functionally, to disrupt the functional interactions of different brain regions, that the orbital cortex needs to interact with the amygdala and the temporal lobe, as well as with the medial dorsal nucleus of the thalamus. Having gotten these results, we were interested in whether there were specializations of function within orbital frontal cortex. The orbital cortex is a huge expansive cortex. I showed you the cytoarchitectonics of delineations of Price and colleagues before, but the truth is there are three main areas. If you remove the subdivisions, there's areas 11, 13, and 14, which are the major subdivisions. And we wanted to investigate whether there were specializations that we could determine within that vast expanse of orbital cortex. From what I told you so far, the deficits after permanent lesions of orbital cortex could reflect an inability to update value. That's how I usually talk about it. I just say there's a failure of value updating. But in fact, it could be more complicated than that. It could be that monkeys with orbital cortex lesions can update the value, but they can't store it. Or it could be that they could update it and store it, but just somehow can't access that information at the time of making a visual choice. So to distinguish among these possibilities, we turn to temporary inactivation studies. So these studies will give us both better temporal resolution and better spatial resolution, and importantly, allow us to disentangle some of these possibilities. The monkeys were trained on the same task as before. This is an evaluation task, but now in an automated format. So monkeys are seeing two-dimensional images on a touch screen. They're making choices on a touch-sensitive screen. We used structural magnetic resonance imaging scans to identify orbital cortex in our monkeys. We infused a compound called FIP, which is a GABA agonist. You may know that's similar to muscimol, but the overall design was the same. And our intention was to inactivate either area 13 in the posterior orbital cortex or area 11 in the anterior orbital cortex. Our hypothesis was that area 13 would be important for this task, and that's because area 13 is the first place within frontal cortex that's in receipt of olfactory, gustatory, and visual sensory inputs. So this seemed like a good and necessary thing to have to link visual information with taste information. So just to be clear, the experimental design involved infusions to inactivate infusions that took place either before selective satiation, in which case orbital cortex tissue was inactive during selective satiation, which is the time when there would be a registration of the new food value. And the other condition was infusions were given after selective satiation, in which case the drug would be in the cortex only during the visual choice test that the monkey carried out. So here are our results for area 13. And instead of different score, we're using proportion shifted. But again, the higher the bar, the better the job the monkeys are doing. So what we found is that if we infused posterior orbital cortex area 13, inactivated it before selective satiation and during selective satiation, the monkeys were impaired on this task. But if we infused after selective satiation, the monkeys were fine. So this indicates that even though the drug is on board here during the choice test, you need area 13 during the selective satiation to register the change in value that happens during selective satiation. But once that's happened, you don't need area 13 anymore. And this clearly indicated a role for area 13 in updating value. For area 11, we found the converse results. Here, infusions before selective satiation had no effect. Infusions after selective satiation disrupted choices. This was surprising. But clearly this indicates a role for area 11 in goal selection. The flip side of goal selection, that is at the time the monkey's making a visual choice, an impairment in goal selection might also be called goal neglect. And indeed there's a long history of study of frontal cortex and goal neglect. Perhaps the best known of these is from Teuber, who tested humans on the Wisconsin card sort task, which requires humans to sort cards according to a color, number, or shape imprinted on that card. And what Teuber reported was that patients with frontal lobe damage could accurately state the prevailing view in a Wisconsin card sort test, but then, in apparent contradiction to that knowledge, would implement the incorrect choice. And these are really stunning if you talk to patients who do this. But again, it's a disconnection of knowledge and action. So what I've told you is that when we tested our monkeys on a test of behavioral inhibition and cognitive flexibility, they actually did quite well. This led my colleagues and I to reject the idea that the overarching function of the orbital cortex is in behavioral inhibition or cognitive flexibility. We also investigated updating outcome valuations using a devaluation task, and that indeed appears to be a true function of orbital frontal cortex. And I hope I've convinced you that the orbital frontal cortex contains distinct functional subdivisions for value updating and goal selection. And one final point is that I don't have time to cover is that similar impairments have been found in humans with damage to the so-called ventromedial prefrontal cortex, lesions which include large sectors of orbital and medial frontal cortex. So one idea that's guided my work is the idea that an understanding of the functions of these prefrontal areas may be gleaned by considering the adaptive advantage conveyed by these areas in our primate ancestors in their time and place. So for the granular orbital frontal cortex, we think this helped early primates. These were small primates relegated to the fine branch niche, and they were foraging in the fine branch niche. And it helped these early primates select and identify foods and identify visual correlates of foods, if you will, if those foods were hidden. And it also allowed them to take into account their current biological states, their state of satiety in foraging. And finally, I'd like to thank all my colleagues who contributed to this work, especially former postdoctoral fellows, Pete Rudebeck, Alicia Izquierdo, and Ben Basil, who all helped with these studies. Technicians in the lab, Don Lundgren, Emma Moylan, and Pingyu Chen. More senior colleagues, Janita Turchi, Richard Saunders, Kay Salim, and Jaewon Huang. Students, Lily Chow and Anna Prescott. And then finally, last but not least, my senior colleagues, Kim Graham, Steve Wise, and Mary Baldwin for ideas and discussions about the evolution of memory systems. Thank you.